Colloquially, we tend to think of peri-implantitis as a kind of periodontitis around an implant. But how accurate is that assumption from a scientific standpoint? That was precisely the question that came to mind after reading a doctoral dissertation from the Harvard School of Dental Medicine, completed in 2026, entitled Multimodal Single-Cell Analysis Reveals Disease-Specific Epithelial-Stromal Regulation of Inflammation in Periodontitis and Peri-implantitis, authored by Yu Stacy, Cho. What makes this research particularly interesting is that it did not stop at the conventional examination of inflamed tissue. Instead, it investigated what happens at the level of individual cells and where those cells are located within tissue affected by the inflammatory process. A total of 20 sites from 20 individuals were analyzed, 5 periodontally healthy sites, 7 sites with advanced periodontitis, and 8 sites with peri-implantitis. The results suggest that tissue surrounding natural teeth and tissue surrounding dental implants behave quite differently during chronic inflammation, which is, to some extent, not surprising. In periodontitis, the author found an increased proportion of less mature cells associated with tissue repair, while at the same time there were fewer mature cells capable of forming a stable protective barrier. In other words, the tissue appears to become trapped in a continuous repair process. Damage is present, the cells are attempting to repair it, but the normal protective structure is never fully re-established. In peri-implantitis, the pattern is different. More mature superficial cells are relatively better preserved, but they show signs of pronounced cellular stress. This suggests that peri-implantitis may not simply be the same disease occurring on a different surface, but rather a distinct biological tissue response. An even more interesting part of the study concerns a signaling molecule known as CXCL14. It was found predominantly in more mature superficial epithelial cells, and in periodontitis, areas with stronger expression of this signal were most clearly associated with the close proximity of immune cells. This raises the possibility that the superficial epithelial layer is not merely a passive barrier damaged by bacteria, but an active participant capable of influencing where immune cells accumulate. In peri-implantitis, this association was weaker, providing another indication that the two diseases differ biologically. However, perhaps the most intriguing finding for me concerns the blood vessels. In healthy tissue, endothelial cells lining the blood vessels mainly perform their usual vascular functions. In periodontitis and peri-implantitis, however, the researchers identified a small population of endothelial cells that began to display characteristics associated with the immune response. These cells were located immediately adjacent to T lymphocytes, which are critically important cells of the immune system. When the researchers exposed endothelial cells in the laboratory to inflammatory signals, they were able to induce a similar change, particularly following stimulation with interferon gamma. This does not prove that blood vessels directly activate T lymphocytes, but it demonstrates something highly important. In chronically inflamed tissue, blood vessels may not merely serve as pathways through which immune cells reach the lesion. They may themselves become active participants in the local immune response. From a practical perspective, this study changes the way we may think about periodontitis and peri-implantitis. Disease is not simply a conflict between bacteria and the immune system. The cells forming the protective epithelial surface, the cells lining the blood vessels, and the immune cells create an interconnected environment that may help sustain the inflammatory process. This opens the possibility that future therapies may not focus solely on biofilm removal and infection control, but also on restoring the normal biological behavior of the tissue itself. The author specifically points toward the potential development of future local therapies targeting either the epithelial barrier or the communication between endothelial cells and the immune system, potentially with fewer systemic effects. Nevertheless, we need to be very cautious. The study is relatively small, there was no healthy peri-implant mucosal control group, the groups were not fully matched for age, sex, or disease severity, and the study was cross-sectional, meaning that it cannot establish what is cause and what is consequence. The microbiome was also not directly analyzed, and the fact that certain cells are located next to each other does not prove that they communicate directly. Therefore, this is not yet a study that will change clinical treatment protocols tomorrow. But it does change the question we should be asking. Instead of viewing peri-implantitis simply as periodontitis occurring around a titanium implant, we may need to accept that it is a disease with its own biological rules. And if that is true, then it is entirely possible that the optimal treatment of peri-implantitis will one day need to differ substantially from the treatment of periodontitis.